and we'll be discussing an important topic of genetics today. So if you can hear me loud and clear, please write in the chat box. Dr. Hassan, is the chat box open? Okay, thank you, Noor Ilma. Okay, Samar, thank you. Okay, so we can't see the shared screen properly. What happened? Can you please elaborate what happened to the screen? Is this happening to all of you?
There is
the
a
screen not?
black box is infrared.
Okay, so it's already 12:05, so I'm starting today's lecture. Now, okay. Discussion is related with the genetics, and as you know that you have been through the genetics before in the previous module of genetics in your you have been through genetics in your first year and second year regularly. So the basic purpose of repeating itself is to reinforce the ideas of genetics, and so that you can incorporate the information of genetics in your practice when you go to your wards of medicine and surgery, or even pediatrics. So today's discussion is just an overview of genetics, in which we will try to go through the basic ideas of the structure of DNA and RNA, and then we'll go through the basic idea of or the definition of genetics. We'll go through the central dogma, what is central dogma and what you really mean by central dogma. Then we will go through the RNA and DNA, the basic structures, the genetic processes. We'll go through the genetic processes like replication, transcription, and translation. So as we all know that it's a big topic today, so it is practically impossible to go in detail of each of the topic. So what I'm going to do is to just try to reinforce some of the ideas, and will go in, not without going into details of each of the processes. We'll try to understand the basic ideas, and at the end you can ask me any queries if you have. So let's start with today's discussion. Genetics. Okay, so what is actually genetics, and what is the purpose of genetics in medicine? Any idea? The chat box is open for you. What is genetics, and what is the purpose of studying genetics in medicine? Is it is it relatable? Can you relate the ideas of genetics with the medicine? Inheritance patterns. Okay, Minham Rataz. Okay, a lot of diseases have, yes. Okay. So inheritance, genetic predisposition of the diseases. Okay, and some we can anticipate diseases in the upcoming. Anticipate. Okay. Okay, so let's start with some basic, very basic definition of genetic. What actually is genetics? Okay, so it is basically a branch of science concerned with genes, heredity, and its variation in the living organisms. So in after the discovery of the structure of the DNA by the Watson Crick in 1953, it was actually a momentous moment in science. And after the discovery of the DNA structure, a new branch of science actually developed, which is known as now as genetics. In genetics, we study the molecular structure and functions of genes. We study the details of processes of trait inheritance from parents to offspring. we study the gene behavior in the context of a cell or organism. So what is basically the gene environment interaction, how the gene and the environment interact itself, and the role of gene environment interaction in development of the disease. Then we study the distribution of the genes, its variation and change in the population. So these are some of the highlights of genetics that what we actually study in genetics. So at this point, what you are actually um, studying is just the some basic levels of genetics. That is, you are uh, studying the molecular structure and functions of genes, and somewhat the process of trait inheritance from parents to offspring, but not actually going into details at this point. But after graduation, if someone is really interested in doing his masters in genetics, it will be really great for it will be really helpful in at this point, that you understand the things, the basic things related with the genetics. Now, how much DNA is actually present in a cell? um, we know that we have a genome. That genome is basically all the composition of DNA and RNA, in which we have approximately, not actually approximately, but we exactly have 46 chromosomes. And genome is basically, our genome approximately consists of 3 billion base pairs. One base pair is approximately uh, 0.000034 meters, and the DNA sequence in any two people is approximate is 99%, 99.9% identical, whereas only 0.1% is unique. So what you need to understand here, that we exactly have 46 chromosomes, and we have approximately 3 billion base pairs, and DNA sequence in almost all the human beings is identical, except 0.1% is unique. So that is what actually we are actually made up of, that we have 46 chromosomes. Now after approximately 50 years of the Watson and Crick DNA structure, in April 2003, the full sequence is published, and researchers determined that within this sequence there was somewhere between 30 to 40,000 genes, and now we believe that there are approximately 25,000 genes we have. So, but what you need to understand here, that we are still not clear about the whole genome. There are some hidden parts, there are some hidden parts of genetics. There are some ambiguities in the processes, in the structures is still left in understanding of the whole genome. So all the processes, they are based on our understanding, but the nature, Allah subhanahu wa ta'ala, it is basically the gift of God, that all the genetic processes are being predetermined and identified by our genome. So how can we use actually this information of genetics? We can use for better understanding of the human diseases. Various genetic diseases are there. There are various inherited diseases. That is, for example, we have a number of diseases which can be inherited, including the cancers. Then we have some enzyme deficiencies. Then we have other neurological diseases, which are all are linked with the understanding of genetics. Then not only understanding of the human diseases is related with the genetics, but other um, things can also be related. For example, now we have a personalized medicine and pharmacogenetics, that the every individual has a unique genome, and according to its genome, the medicine is being given to that specific individual. Then it has a role in cognitive function. So the understanding of genetics can help in getting the insight of the cognitive functions. Then the origins, and in understanding of the human origins, and identifying genetic susceptibility to the disease, as some of the student identified it correctly, that we can identify genetic susceptibility to the disease, for example breast cancer diseases, for example hypertension, for example diabetes mellitus. So we can understand the role of genetics in identifying genetic susceptibility to the disease. Now, coming to the three important function of genes, or the nucleic acids. Nucleic acids are of central importance in the storage, transmission, and expression of the genetic information. So genetic information is actually stored, transmitted, and expressed in the form of a nucleic acid. Nucleic acid, there are two types of nucleic acid, that is RNA ribonucleic acid and the deoxyribonucleic acid. They play an important role in the storage, transmission, and expression of the genetic information. Now, how these three important functions they are being performed. Storage is basically all the genetic information is actually stored in the form of a DNA in a double helical structure, in which the sequence of the nucleotides are there, and it is basically the storage form of the genetic information. Then how it is being how it is being transmitted. It is actually transmitted um, from one generation to another uh, through the replication process, in which the exact copies of the DNA are being formed. And the third one is the expression. So the nucleic acids they express themselves by formation of proteins. So the expression of nucleic acids is basically the formation of proteins, and the formation of proteins it's known as translation process. So the DNA expresses itself through translation by forming the proteins. Now, the coming to the central dogma of molecular biology. Central dogma of molecular biology is is a principle or is a hypothesis that says that the genetic information flows from the DNA to the RNA and then to the proteins. So there are three processes which are involved in this transfer of genetic information. It is replication, transcription, and the translation processes. So the central dogma of molecular biology states that the genetic information is transferred from the DNA to the RNA and then the formation of proteins. So there are three processes which helps in the transfer of genetic information. We have replication process, in which the DNA exact copies, Xerox copy of DNA are being synthesized. Then transcription, through which the RNAs are being synthesized from the DNA, and then the translation process through which the proteins or polypeptide chains are being synthesized. Another diagrammatic representation, flow diagram of the central dogma of molecular biology, that is the genetic information is being transferred from the DNA to the RNA and to the proteins. Now, coming to the nucleic acids. Nucleic acids are there are two types of nucleic acids. We have DNA and RNA, and they are made up of nucleotides, which are also known as the building blocks of the nucleic acids. Now, these nucleotides are they are formed by nucleosides and phosphates. Then nucleosides, they are further consist of nitrogenous bases and sugar, whereas nitrogenous bases there are two families of nitrogenous bases. We have purines and pyrimidines, and we have two types of sugars. In purines, we have adenine, guanine, whereas pyrimidines contain cytosine, thiamine, and uracil, whereas there are two types of sugars. We have ribose sugar in the RNA and deoxyribose sugar in the DNA. So basically, nucleic acids are made up of nitrogenous bases, two types of nitrogenous bases, purines, pyrimidines, and sugar. It is ribose and deoxyribose sugars. Now, how they are actually binded with each other. For example, we are taking an example of a nucleotide in which we have the ribose sugar. Then a nitrogenous base is represented by a blue square, and then we have a phosphate represented by pink circle. and then we have a phosphodiester linkages that binds the two nucleotides with each other. So the second nucleotide is here, and the hydroxyl group, which is present at the 3' position, binds with the phosphate, which is present at the 5' position, forming a phosphodiester linkage. Now this phosphodiester linkage uh, binds the nucleotide with each other and forms the backbone of the DNA. So in this way, the nucleotides are binded with each other with the help of the phosphodiester linkages, and the backbone is formed with the help of the phosphate ribose, whereas the nitrogenous bases are stacked in the center of the DNA. So the backbone of the DNA is actually made up of the phosphates, ribose sugars, phosphate, ribose sugar, and the phosphodiester linkages are binding the backbone of the DNA running from the 5' to 3' position. Now, these are the two guys who are responsible for the whole genetic module. So, so these are the two guys who actually discovered the DNA structure and some features of Watson-Crick model of DNA as you have done it before. As you can see on the diagrammatic representation, we have the vertical arrow and the horizontal arrow showing the width of the DNA molecule. Approximately the width of the DNA molecule is 20 degrees Armstrong, whereas the height of the complete turn of a DNA molecule, that is 360 degrees turn. After complete turn, the height of the DNA molecule is 34 degrees Armstrong. It's a double helical structure in which the nitrogenous bases are stacked in the center with the help of the hydrogen bonds, and the backbone is formed by the phosphate sugar, phosphate, ribose sugar in this way, and they are helically wounded around the central axis, which is represented by the dark black line. So it's a central axis of DNA along which the DNA is being twisted around, and through while making these helical structures, their two grooves are being formed, that is minor groove and the major groove, which binds with the regulatory proteins. Coming to the RNA are unbranched single-stranded polymeric molecules composed of nucleoside monophosphates joined together by the phosphodiester bonds. So they are unbranched single-stranded polymeric molecules, unlike DNA. As DNA is a double helical structure, the RNA is a single-stranded polymeric molecule. There are three major types of RNAs that are participate in the process of protein synthesis. We have ribosomal RNA, transfer RNA, and the messenger. These are the three major types. However, we have some other types of RNAs as well. For example, we have a small nuclear RNA, we have heterogeneous RNA as well. But there are the three major types, that is ribosomal, transfer, and the messenger RNA. So by the transcription process, through the transcription process, not only messenger RNA is synthesized, but other RNAs are also synthesized. It is transfer RNA, the ribosomal RNA, and the messenger RNA is also being synthesized. All these three types of RNAs, they have three different functions. We have prokaryotic ribosomal RNA. The ribosomal RNAs are those RNAs that compose the ribosomes with the help of the ribosomal proteins. There are three species of prokaryotic ribosomal RNAs, whereas there are four species of eukaryotic ribosomal RNAs. So the basic purpose of the ribosomal RNA is to form the ribosomes with the help of the ribosomal proteins. Then we have transfer RNA. The basic purpose of transfer RNA is to transfer the amino acids or activate the amino acids, transfer the amino acids from the cytoplasm and takes it to the surface of the ribosome, where the translation process occurs. So the transfer RNA, it is a clover leaf-like shape. It has two ends, 3' and the 5' end. At the 3' end, the sequence ACC that binds, as a specific sequence is there that binds to the specific amino acid. And then we have the pseudouracil arm, we have the anticodon arm, and we have the D arm that have different functions. So without going into detail, we'll proceed to the messenger RNA. Now, the messenger RNA, as the name suggests, the it helps in decoding the message. As you can see, it's just a diagrammatic representation taken from your booklet and card. It has a 5' end at which it is being capped with the help of the 7-methyl guanosine triphosphate. It is a cap structure. It is an unusual triphosphate linkage, backward linkage, 5' 5' linkage, unusual linkage, and it is just to protect the messenger RNA from the nucleases enzymes. Similarly, we have the structure of poly A tail at the 3' end. A long adenine residues are being added at the 3' end, and also this structure helps in the protection of the messenger RNA from the nucleases enzymes, as well as it also helps in the transfer of messenger RNA from the nucleus to the cytoplasm where the translation process occurs. In the center, we have the coding region that contains the codons, and these codons then help in the synthesis of the polypeptide chain. Now, we have genetic processes. We have 3 basic genetic processes that helps in the transfer of information from the DNA to the RNA and then to the protein synthesis. Just before jumping on to the 3 processes, what you need to understand here is just the alignment of the DNA and RNA. As we know that DNA is a double helical structure, 2 strands. Both the strands are anti-parallel and complementary to each other. Anti-parallel, that the ends are opposite to each other. If this strand is running from 5' to 3', the other strand will run from the 3' to 5' position. The one strand will be known as the coding strand, and the other strand will be known as the template strand. Now, through the transcription process, the RNA polymerase enzyme will read the template strand from the 3' to 5' position. It will read it in this direction, and it has a characteristic that it can only read from 3' to 5' direction and synthesizes the messenger RNA. That is a premature messenger RNA is being synthesized from in the anti-parallel and complementary strand. So the template strand is being read by the RNA polymerase enzyme, whereas the coding strand is exactly the same sequence as that of the pre-messenger RNA, a newly synthesized messenger RNA, except the thymine is replaced by the uracil. So the ends are same with the coding strand. That is why we call this strand as a coding strand, and this red represented by red is known as the template strand. Now, after formation of the newly formed messenger RNA, it is premature RNA. This immature RNA will go into the maturation process, which is known as the post-transcriptional modification. Through post-transcriptional modification, the cap is being added, the tail is being added, and the introns are being removed in the eukaryotes. So after the maturation, it will go for the translation process, where the mature messenger RNA will help in the translation process at the ribosomal surface. At the ribosomal surface, the mature RNA will contain specific codons. The first codon will always be AUG. So AUG will be the first codon always. It will read the codons, and the translation process occurs, and a polypeptide chain will be synthesized from the amino terminal to the carboxylic terminal, and various amino acids are being incorporated according to the codons in the polypeptide chains. So this animation will help you understand the alignment of the DNA, the messenger RNA, and the polypeptide chains. Now, coming to the first process of replication. Replication is formally defined as production of multiple identical copies of a DNA molecule by binding the 2 strands of the DNA double helix and forming the new complementary strands. So is basically replication is a process in which the identical Xerox copy of the DNA is being synthesized. However, as we know that we have a limited time of 1 hour, so we will not go into detail of into different prokaryotic and eukaryotic replication processes, but we'll just try to touch and go with the basic ideas of the replication process. So in prokaryotic replication, the prokaryotic DNA is in a closed circular structure. So after the replication process, it will go into each daughter cell. So each DNA will go into each daughter cell. Whereas in the eukaryotic replication, it is the open-ended thread-like structure. So after the replication process, each DNA molecule will go into the each daughter cell. So what you need to remember here, that the prokaryotic DNA is a closed circular structure, so its replication is a little bit different from the eukaryotic replication process. Now, the requirements which are required for the replication process, for replication, as the DNA is being synthesizing, so we'll require an enzyme that will synthesize the DNA, and this enzyme is known as the DNA polymerase enzyme. We have three basic types of DNA polymerase enzymes. In the prokaryotes, DNA polymerase 1, that will the basic function of polymerase 1 is RNA primer removal and replacement with the DNA. Then we have polymerase 2. The basic purpose is to proofread and repair the DNA structure, and polymerase 3 is the synthesizing of the leading and the lagging strand and proofreading as well. So we have three different types of enzymes: DNA polymerase 1, 2, and 3 in the prokaryotes, and these are some of their important functions. Whereas in eukaryotes, there is a single DNA polymerase enzyme containing subunits like alpha, beta, gamma, sigma, and epsilon. These subunits have different functions. For example, alpha subunit has primase like activity. It will synthesize primers. It will initiate DNA synthesis on both the on the lagging strand. Beta subunit will help in the repair of the DNA structure. Gamma replicates the mitochondrial DNA, and sigma elongates both the leading and the Okazaki fragments, and epsilon will help in the repair of the DNA structure. Some other proteins which are being involved in the synthesis of DNA are single-stranded binding protein that helps in the reassociation of both the strands. It keeps both the strands apart from each other and also protects it from the nucleases enzymes. Similarly, DNA gyrase enzyme, it's a topoisomerase 2 that relieves the torsional strain, which is being generated by the unwinding of the DNA. So the DNA gyrase enzymes, there are two types of topoisomerases. We have topoisomerase 1 and we have topoisomerase 2. The topoisomerase release the torsional strain by temporarily breaking and resealing single strand, whereas topoisomerase 2 it releases the torsional strain by temporarily breaking and resealing both the strands. In E. coli, the topoisomerase 2, the name of the topoisomerase 2 is this DNA gyrase enzyme. Then we have a DNA ligase enzyme that joins the Okazaki fragments of the on the lagging strand. So these are some of the proteins or enzymes which are involved in the DNA synthesis or in the replication process. Now, replication process is a continuous process, but for our easiness, we have divided the replication process into steps. These steps are human-made steps just for the understanding of the process. In nature, there is no steps. Okay. However, there are sequences. There are sequences of processes. So we have identification of origin of replication. We have unwinding of double-stranded DNA. We have formation of the replication fork, initiation of DNA synthesis, and elongation. We have formation of replication bubbles, and we have the reconstitution of the chromatin structure. So these are some of the steps of the replication process. Starting from the identification of origin of replication, the origin of replication is being identified by OREC, and then the double strands are being unwinded, then the replication forks are formed, then the DNA is synthesis being initiated on both these strands, the leading and the lagging strand, and then the formation of the replication bubbles, and the last step is the reconstitution of the chromatin structure. So the origin of replication, that is OREC, is being identified by the ORI binding protein, and this origin of replication, first, as it is being identified, the AT rich region just before the origin of replication is being pulled apart with the help of the helicase-like activity that breaks down the hydrogen bonds between the two strands, and immediately they are being binded with the single-stranded binding protein that keeps both these strands from reassociation. Not only from reassociation, but also protects it from the nucleases enzymes. Ramesh, can you please switch off your camera? Ramesh. Okay. So so that's the origin of replication, in which the origin of replication is being identified by the ORI binding protein, and then the single-stranded binding proteins, they keeps both the strands apart and protects it from the nucleases enzymes. When both the strands are pulled apart with the helicase-like activity, the helicase will break the hydrogen bond between the two strands, and through this, the replication forks are being formed on both ends of the replication bubble. So in this way, a replication bubble is being formed. At each end, we have a replication fork. At each end, we have one leading strand and one lagging strand. So this is a bidirectional process. The replication is a bidirectional process, and as we know that in prokaryotes is a closed circular structure. So after synthesizing the whole DNA, the replication bubbles will fuse and will form two different daughter DNA. Now, taking an example on each side of the replication bubble, as this is the direction of the movement of replication fork, so there will be a leading strand which runs from the 3' to 5' position. This is known as the leading strand. The reason behind only is that the DNA polymerase enzyme can read the leading strand from the 3' to the 5' position and can synthesize the new strand from the 5' to 3' direction. So this is why this is known as the leading strand. The leading strand, the DNA is synthesizing in one complete DNA. The whole long DNA, complete DNA is being synthesized. Whereas on the other side, this side is known as the lagging strand. The reason behind is that it is opposite to the direction of the movement of the replication fork, and it in the opposite direction. And the reason again is that the DNA polymerase enzyme can only read the strand from the 3' to 5' direction.
The two parent strands of DNA are antiparallel. They are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
So I hope you have understood the whole continuous process of replication after watching the video, the animation video. So we are now moving to the transcription process. Now, transcription again is simply transcription is a synthesis of RNA from DNA, and it is formally defined as the process of RNA synthesis directed by the DNA template is called the transcription process. Now, many of the students, they have a misconception that only messenger RNA is being synthesized by the transcription process, but in real, all types of RNAs are being synthesized through the transcription. But for our easiness, we always explain the messenger RNA synthesis just to keep it more simple. So the requirements for transcription are RNA polymerase enzyme, the RNA polymerase that synthesizes it, polymerizes the RNA nucleotides, and the nucleotide triphosphates as substrates, the DNA as a template. So we are going to have two template strand and the coding strand. So these are some of the requirements of the transcription process. Now, as we are keeping it simple, and we will be comparing some of the eukaryotic and prokaryotic transcription processes. So if we compare with prokaryotic and eukaryotic, they are simple. The comparison between the enzymes, the single RNA polymerase, we have a single RNA polymerase enzyme in prokaryotes, whereas we have three different types of RNA polymerases in eukaryotes. We have RNA polymerase 1, 2, and 3. RNA polymerase 1 synthesizes ribosomal RNA. Polymerase 2 synthesizes the heterogeneous RNA or messenger RNA, and 3 synthesizes the transfer RNA and only the 5S species of the ribosomal RNA. Whereas in prokaryotes, there is a single RNA polymerase enzyme containing 2 alpha subunits, 1 beta and 1 beta prime. Whereas in prokaryotic transcription, we require a sigma factor for initiation, whereas there is no sigma factor. Instead of sigma factor, we have transcription factors. For example, we have transcription factor 2D. Then for termination, in prokaryotes, we have Rho protein, whereas in eukaryotes, the Rho protein is not there and is not being understood in detail. Whereas prokaryotic transcription is inhibited by rifampin, actinomycin D, whereas in eukaryotes, the ribonucleic RNA polymerase enzyme 2 is inhibited by alpha-amanitin, which is a mushroom poison, and actinomycin D, which is a chemotherapeutic agent. So this is just some simple comparison between the prokaryotic and eukaryotic transcription processes. Now, just before jumping to the transcription process, it's really important to understand the transcription unit. The transcription unit, as we know that we have a DNA that has 2 strands, the template strand and the coding strand. The template strand runs from the 3' to 5', whereas the coding strand runs from the 5' to 3' direction. The code, the nitrogenous base, which is being transcribed first, is given the number +1, and then we have the +10 regions, and then we have minus regions. In the minus region, we have promoter sequences, and then we have the coding region here on the plus directions. So the DNA strand from +1 to the to termination is known as the transcription unit. So the start site to the terminator is known as the transcription unit, and this transcription unit is being transcribed and synthesizes the RNA. It can be messenger RNA. So this is the transcription unit. Transcription unit is from the start site to the termination side of the transcription. And on the right side we have the downstream, and on the left side we have the upstream where the promoter regions are there. In prokaryotes we have different promoter regions. In eukaryotes we have different promoter regions. In prokaryotes we have box, which is present at -10 region, whereas we have -35 sequence. They are consensus sequences and they work along with each other. So both the promoter regions are required for the functioning of the promoter regions, the box and the -35 sequence. Whereas in eukaryotes, we have got Tata box, which is actually also known as the Hogness box and present at the -25 region. And whereas we have a cat box, which is present at the -70 region. Again, they are consensus sequences and they work along with each other. Similarly, just like start site, just before the start site, we have promoter regions. Similarly, we have terminators as well. In prokaryotes, we have Rho independent types of termination in which the hairpin-like loop is being formed, which is GC rich, guanosine cytosine rich hairpin-like loop, which is followed by the 6 to 8 uracil residues. Similarly, another type of termination is Rho independent, where Rho dependent, whereas the Rho protein is being required for the termination process. In eukaryotes, again, there may be some combination of both the Rho independent or dependent, or whether it's not fully understood. So we will keep our discussion only to the prokaryotic terminations. In prokaryotic transcription, it's a little bit different from it's a little bit different. As you can see here, the prokaryotic transcription unit starts from the start site to the termination site, and we have some promoter regions at -10, -35 regions, and then we have in the center we have the coding region, and then we have some untranslated regions are also there. So the coding region is there. So after the transcription, the whole messenger RNA is being transcribed, containing the 5' region and the 3' region. Or the 3' region, we have the specific region which also have a Schandel-Gano sequence as well. In the center, we have the coding region, and then it is followed by the GC rich, and then the uracil residues. So these are the 3' untranslated regions, and this is the 5' untranslated regions, which are not involved in the translation process of protein synthesis. 5' untranslated and 3' untranslated regions, and then these coding regions will help in the synthesis of the polypeptide chains. Coming to the eukaryotic transcription, again, the difference is between the promoter regions, different promoter regions, different we have poly A tail signal here, and then we have introns and exons, which after maturation these introns are being removed. So this is a product of the messenger RNA, and as you know that the exact copy of the coding strand. So the messenger RNA is exact copy of the coding strand. So it has copied all the same except the thymine is replaced by the uracil. So the introns are there; they are being removed after the post-transcriptional modification. So prokaryotic RNA polymerase enzyme, sigma factor. We have a sigma factor that recognizes the promoter regions, and then we have 2 alpha subunits, 1 beta prime and 1 beta subunit. They have different functions. RNA polymerase enzyme performs following function. It recognizes the promoter region. It helps in the separation of the DNA with the help of the helicase-like activity and topoisomerase that release a torsional strain. Then we have RNA polymerization, and then the recognition of the termination sequence. These are some of the functions which are being performed by the RNA polymerase enzyme. And then eukaryotic RNA polymerases, as we have discussed, they help in the synthesis of different types of RNAs. Stages of transcription. For our easiness for understanding the transcription process, we have divided into stages. For example, the first stage is the transcription complex formation, initiation, elongation, and termination. It is being regulated at the first stage and the termination stage. These both are the control points, the transcription complex or the template recognition, and the termination. They are the control points or the regulatory points of the transcription process. Now, some general model for prokaryotic transcription. As we know that we have promoter regions, the sigma factor recognizes the promoter sequence and binds with the DNA. Both these strands are being unwinded, and during these unwinded regions, the newly incoming RNA nucleotides, they will form the hybrid between RNA and DNA bonds, and along with this, the topoisomerases are also there that are relieving the torsional strains during the unwinding of the DNA structure. The new messenger RNA is being synthesized, and in prokaryotes, the transcription and translation process occurs simultaneously. So during the transcription process, the translation can also occur at the same time. When it reaches the termination site, the GC-rich hairpin-like loop is there that destabilizes the transcription complex, and the new messenger RNA is being released in the cytoplasm. In eukaryotes, we have transcription factors that identifies the promoter region. After it is being identified, both the strands are being separated by the helicase-like activity. The RNA polymerase enzyme binds with the DNA and the transcription factors, forms the transcription complex, and it initiates and elongates the messenger RNA. While forming the messenger RNA, the RNA polymerase enzyme reaches the termination site, and there are two types of terminations, the Rho independent and Rho dependent. The Rho is a protein. In Rho independent, where the Rho protein is not being required, when the RNA polymerase enzyme reaches the termination site, the GC-rich hairpin-like loop is formed that will destabilizes the transcription complex, and all the constituents of the transcription complex, they are being separated from the transcription complex so that they can be used again, and the newly synthesized RNA is released in the cytoplasm. Whereas in Rho dependent, a protein is required. The Rho dependent is a protein that is binded with the RNA, the messenger RNA. When the RNA polymerase enzyme reaches the termination site, it slows down, but the Rho protein does not, and hits the transcription complex, destabilizing the transcription complex, and this complex is being dissociated, releasing the new messenger RNA in the cytoplasm again. Now, this is the animation which can help you, which will further help you in understanding of the transcription process.
In bacterial cells, the transcription of a gene or group of adjacent genes called an operon is controlled by a set of proteins interacting with specific sequences in the DNA molecule, leading to the initiation of transcription and the termination of a transcript. Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site. The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule. By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding of the Rho protein to the RNA. It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase. Between the Rho recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription, resulting in the full-length mRNA transcribed from the gene. In bacterial cells, the transcription of
So we have different hypotheses of terminations of transcription. It can be Rho dependent, it can be Rho independent, or it can be a combination of both the Rho protein and the formation of the GC rich hairpin-like loop. Now, coming to the translation process, the last process of translation, it is formally defined as a process of protein synthesis directed by the RNase is called the translation. So it is not just simple protein synthesis, but it is actually directed by the RNase, not only messenger RNA, but in this process, the transfer RNA, the ribosomal RNA are also being involved. So where the protein synthesis occurs, the protein synthesis occurs at the ribosome, which is our machinery that synthesizes the proteins. In ribosome is actually made up of the two subunits, the larger subunit and the smaller subunit. So in prokaryote, we have 50S subunit and 30S subunit. These subunits are made up of the ribosomal RNase species and ribosomal proteins. Similarly, eukaryotic ribosomes have two subunits, which are different, little bit different from the prokaryotes. We have 60S larger subunit and we have 40S smaller subunits. Now, these are the functional side of a ribosome. As you can see, the smaller subunit, the larger subunit, and then we have some functional sites. We have the exit site, E site, P site represents the peptidyl site, and the A site represents the amino acyl site. So E site where the amino acid is being removed, whereas the P site is where the peptide bond is being formed, and the amino acyl site is where the newly incoming amino acid is being incorporated. Now, for this, what you need to understand is the genetic code. Genetic code is a sequence of 3 nitrogenous bases, and we have 4 different types of nitrogenous bases. So the 3 sequence of nitrogenous bases makes one codon. So according to this, we can have 64 different codons. So as you can see here, the first nucleotide, the second nucleotide, and the third nucleotide. So a combination of these nucleotides, for example, if we have the UCA, we're going to have the amino acid leucine. Similarly, methionine is the methionine is here, which is the first amino acid. And so in this way, we can have the 64 different codons. Out of 64, we have 61 codons that codes for amino acids, and we have 3 codons which are known as the termination codons. Stages of translation process: we have activation of amino acids, we have initiation, elongation, termination, and the post-translational modification. So first, the amino acids are being activated, which are present in the cytoplasm. They are being activated, they are binded with the transfer RNA, and then the transfer RNAs will transfer the amino acids from the cytoplasm to the ribosome. Then the initiation, then elongation, termination process occurs. Now, just the representation of the messenger RNA and the transfer RNA. The codons are in the messenger RNA. For example, we have the codon AUC, and the direction is from 5' to 3' direction. Whereas the anticodons, which are present on the transfer RNA, they are so they are anti-parallel, as you can see, 3' to 5'. They are carrying the specific amino acid according to this codon. So the transfer RNA binded with the specific amino acid binds with the codon in the anti-parallel and complementary way, and in this way the amino acids are being added to the polypeptide chain. Now, this is the first amino acid which comes at the peptidyl site, carrying the formylmethionine in the prokaryotes. The specific transfer RNA is carrying the formylmethionine. The anticodon is UAC that binds with the first codon, which is AUG. So the anticodon and site of the ribosome, and this messenger RNA will align itself with the smaller subunit because of this Shine-Delgado sequence. The Shine-Delgado sequence is there that aligns the messenger RNA with the exact site or peptidyl site. The AUG will occupy the peptidyl site. So in this way, the messenger RNA will bind with the smaller subunit of the ribosome. Now, here we have the animation that will summarize the whole process of translation.
Translation initiation is a process in which mRNA, initiator tRNA, and small and large ribosomal subunits associate with each other to form a complex. Initiation of translation in bacteria begins when the 30S or small ribosomal subunit binds to the mRNA near its 5' end. This process is facilitated by a Shine-Delgado sequence in the mRNA, which is complementary to a component of the small ribosomal subunit called 16S ribosomal RNA. Though a shorter sequence is shown here, the Shine-Delgado sequence is actually 9 nucleotides long. Initiation factor 3 also facilitates the binding of the mRNA to the small ribosomal subunit. Once the 30S subunit IF3 mRNA complex has formed, initiation factor 2 binds to the complex and promotes the binding of the initiator transfer RNA for N-formal methionine to the complex. IF2 and IF3 hydrolyze GTP as an energy source to promote the association of mRNA, tRNA, and the ribosomal subunits. Translation initiation is completed when the large ribosomal subunit binds and IF2 and IF3 are released. Later, when translation is completed, IF1 is needed for the dissociation of the complex. Chain elongation begins with the binding of a tRNA, which recognizes the next codon in the mRNA to the A site of the ribosome. This is catalyzed by the EFTU transcription factor and requires the hydrolysis of a GTP. Once the tRNA binds in the A site of the ribosome, The polypeptide chain is moved from the tRNA in the P site to the amino acid attached to the tRNA in the A site. Peptidyltransferase, a protein RNA complex present in the 50S ribosomal subunit, catalyzes the formation of this new peptide bond between the amino acids. The ribosome then translocates to the next codon. This process is promoted by elongation factor G and requires another GTP. This places the empty tRNA molecule in the E site of the ribosome and moves the tRNA containing the growing polypeptide chain in the P site. The next codon in the mRNA chain is positioned in the A site. The uncharged or empty tRNA in the E site then leaves the ribosome and a cycle of chain elongation is completed. Through subsequent cycles of chain elongation, the polypeptide chain continues to elongate one amino acid at a time. Termination begins when a stop codon appears in the A site. Since there is no tRNA corresponding to the stop codon, a release factor binds in the A site. The binding of the release factor causes the polypeptide chain to be cleaved from the tRNA. The polypeptide is released, and then the tRNA is released. In the last step, the two ribosomal subunits and the mRNA dissociate from each other. This completes the termination process. Translation.
Okay, so we have understood the translation process, and if we have various ribosomes attached to the same messenger RNA at the same time, synthesizing various copies of polypeptide chains at the same time, this is known as polysome. So in this way, this is the efficient way of utilizing the messenger RNA, which is being utilized by multiple ribosomes, and we call it as a polysome. Now we have reached to the end of our discussion, and just for your information, the animations they have been taken from the YouTube. The channel name is Magro Hill Animation, so you can go to this channel and find out the animation videos of replication, transcription, and translation process. Is very easy to understand through these animation processes. So if you have any queries, please write in the chat box. I'm waiting for your queries. If you have any queries, please write in the chat box. What are Okazaki? Oh, here you go. We can. Okay, Priyanka, what are Okazaki fragments? Okay, Okazaki fragments are discontinuous fragments of newly formed DNA on the lagging strand. As we have discussed that on the replication fork, on each side of the replication bubble, we have the replication fork. So as the direction on each strand, both the strands are involved in synthesizing of new strands. So one strand will be towards the direction of the replication fork, whereas the other will be opposite to the replication. So in the opposite direction, there will be discontinuous synthesis of DNA fragments, and they will be known as the Okazaki fragments. They will be binded to each other with the help of the ligase enzymes and will form the continuous fragments later on. Minha Murtaza, are new amino acids added at the A site or the P site? The new amino acids are added at the A site. But what you need to remember here, that the first amino acid occupies the P site. The formyl methionine comes at the P site, and then the new amino acid, they keep on coming at the amino acyl site. Okay. Okay, so all the information which you have seen in this presentation is being taken from Lippincott and Harper. So the reference textbooks, recommended reference textbooks are Lippincott and Harper for this lecture. So in this lecture, just to summarize our today's discussion, this discussion was about the overview of genetics. We have discussed it, we have formally defined genetics, what is genetics, and how we can use the information gained from genetics. Then we have revisited the ideas of the structures of DNA and RNA and the three processes we have went into details of the three processes like replication, transcription, and translation. So I think we are done with today. Dr. Hassan, are you there?
Yes. Can we end the meeting?
Yes, please log me out, and thank you for hosting this meeting.